You know, one of the great pleasures of DPUK is seeing the development of young scientists and also not so young scientists. Now is the time in the program where we celebrate the winners of the poster competition. So first of all, congratulations to all of you who supplied posters. I think that, you know, well, the diversity and the detail, the complexity of some of these posters is very encouraging. I'm sure I didn't understand most of what I read, but hey, there are, don't worry about that. So let's go to the runner-up. Okay, the runner-up. I would wonder whether Roche Diagnostics, Ashton Harper would like to come up. Hello, Ashton. Okay. So we're very grateful for Roche Diagnostics sponsoring this prize, and the prize will be presented to the runner-up. And the runner-up is Dr. Delia Georg from the Transylvania Institute of Neuroscience. Delia. Whoa.
Oh, sorry.
We have...
Are you?
Oh, well done. Congratulations. This is the point where the TV cameras go to different early career researchers for the dramatic effect. So the winner is Elijah Mack from the University of Cambridge. Please, Chairman. And we're really grateful to ARUK, and this is Shirna Scales, who's going to present the prize on behalf of ARUK.
Bear with us a second.
Congratulations. Okay.
Congratulations, it was really good.
So whilst Vanessa comes up to introduce the next session, would you give a round of applause to our winners?
So we're on the homeward stretch for our conference today. I think it's been a really good couple of days so far. We've been hearing an awful lot about exciting developments around biomarkers. I think that's one of the key things that's a dramatic change that's happening in our field at the moment, and also the possibilities and maybe cautions we need to have around the use of big data and AI. This session is going to be about trials, and it's a little more focused on the implications for our patients and the wider population at scale now. I tend to think of myself as a clinician who does research, and I've been working in this area in different ways for the last 30 years. It truly is one of the most exciting times, I think, at the moment in terms of development, as I said, around biomarkers, but also new treatments. And I feel an awful lot of hope at the moment for what I'm able to offer my patients. We heard a very moving story from Chris this morning about the reality of living with memory impairment. And this is such an urgent area of need. I'm just tremendously grateful for everyone that is contributing to this area of research and very excited about the possibilities moving forward. So this afternoon is Trials for All. So as I said, strong focus on how we provide real change for real people very quickly. So I thought I'd dwell a little bit on what we need at this point. There's been a lot of changes in the UK over the last couple of years in terms of dementia research infrastructure. We have the Barbara Windsor Dementia Mission, which is a piece of government-funded infrastructure, and we're going to be hearing more, I'm sure, about that from Hilary shortly. And then at an event at number 10 a couple of weeks ago, we had the announcement also of a Dementia Trials Accelerator to help augment and accelerate some of the work the mission is trying to do. And this has been really about trying to bring some of the enthusiasm and speed that we saw in COVID when we were able to create vaccines at scale and say, how can we use that type of engagement, enthusiasm, power to address this huge unmet need in dementia? As part of the dementia mission, there has been a huge amount of funding going in to create a trials network under the DTRC, the Dementia Translational Research Collaboration Group that already existed, and I'm involved in that, I'm pleased to say. And then there's reconfigurations of other things going on in the UK. So our clinical research network, which has been around for a good number of years, supports and facilitates research across sites across the UK, is being relaunched this year as the Research Delivery Network. So we're waiting to see with what implications that has on access and the type of research that we're able to support. And then Dementias Platform UK, of which I'm one of the associate directors, is going for renewal as well at the end of next year. So there's a lot of changes, and I'm hopefully going to give you a little bit of a flavour of what we've been doing in DPUK around trials. So despite all of these changes in infrastructure, I think we're still very aware that there's a lot of work to do in order to be able to make our trials as available to patients as possible and to be able to subsequently deliver real change to care in clinics. They need to be faster. They need to be set up more efficiently. We need to find a way, frankly, to get over some of the bureaucracy I think that we have a lot of in the UK in terms of regulation and setup. Or how do we make it work more effectively, put it that way. And the other thing we've heard quite a lot about already is the need to diversify more in terms of the populations that we recruit into research. These huge possibilities around things like plasma biomarkers are only going to really work if we can get data from a much more representative population than we do now. And I think you can say that in terms of a lot of the issues we've discussed, including AI work as well. And also, I think, we're Dementias UK. We're very focused on UK infrastructure. But actually, we are an island, but we're not an island. We should be doing this with partners internationally as well. And I hear a lot from academics and industry partners, the fact that what they don't want to be doing is going to individual countries and working with individual cohorts, registries set up. We need to be thinking wider than that. So from a DPUK perspective, we're trying to think, how can we align ourselves with partners beyond the UK? So the part of DPUK that I oversee is something called the Trials Delivery Framework. This has been built on over the last couple of years. In the initial funding of DPUK, it was very much focused on our cognitively healthy and preclinical registers that Ivan Koichov leads, and those have been really growing at pace and in terms of the level of characterization. But then over the last couple of years, we've built a really diverse, large network of clinical sites that I think is probably now the largest dementia-focused and certainly the most diverse network in terms of the sites we represent and therefore the populations that we can offer research to. And I'm going to just tell you a little bit about a new project that we've just got funded to do a large amount of sampling in those populations around blood and also CSF. So hopefully we can build on that in order to be able to characterize people at our sites in our cohorts so we can get them into trials as effectively as possible, get those trials giving us really good data that we can then deliver to patients. So just to summarize, I'm not going into detail on this, but it just gives you a flavor of the extent of our cognitively healthy preclinical registers. We have about 60,000 people sitting in our clinical studies register, and because these people are pulled from existing UK cohorts, we have a really large amount of lead-in data. I mean, you can see there 22,000 people with genetic data within that cohort. And then on top of that, we've built an additional cohort, Great Minds, where people come in and have serial cognitive testing. A proportion are undergoing a genotype actigraphy to look at things like gait and how useful those are for predicting risk. And these are led also by, as I mentioned earlier, by Ivan Koichov. So really building up at scale, and we've got 26 studies running through these registers as a result now. But then, as I mentioned, we've worked really hard over the last couple of years to expand a clinical network of sites. We now have 52 sites across the UK. In addition to the 52, we've partnered with NHS Research Scotland. They have 14 sites north of the border, representing 14 Scottish health boards. So collectively, that means we've got 66 sites across all the nations in the UK, which means now effectively we have access to populations that sit within a quarter of all NHS trusts and a quarter of GP practices. And that's because we've also consciously gone out to start working with primary care networks. So we have two key partners that we are working with and hopefully going to do more work with in the future. And I think this is because, again, as an old age psychiatrist, I'm very aware that 20 years ago, or 30 years ago, when I first did my first memory clinic, I used to see people for follow-up appointments. And that was actually very nice because I got to develop a relationship, I got to develop a level of engagement with the person, and you felt you were somebody that they could come back to if they had questions or concerns or they wanted to get involved in research. Sadly, most services are not able to do that now. And so for lots of patients, they're seen once or twice in secondary care, they go back to primary care. So if we're going to be able to offer research truly to everybody and gather that diverse data we need, we're going to have to do it in primary and secondary care. So that's why we've consciously expanded into that group. And we've also been setting up a clinical registry across our sites that will collect data around dementia diagnosis, risk data around things like cardiovascular health and ApoE status. And we're starting with 9 of our sites and now are going to be expanding into primary care. And because of the great possibilities with our data portal that Simon talked about this morning, we're going to be looking to do live NHS data linkage through that registry and then looking beyond the UK. So we have a DPUK Australia, we obviously have strong links with Korea, and we're also looking to potentially align what we're doing with a new registry initiative called InRAD, which you'll hear more about this afternoon from Robert Perneczky. And because we're DPUK and we already support a lot of existing studies, hopefully we're able to align some of the data from those studies in with that registry. So I'm going to talk to you about one of those particularly called Readout. But what I'll just highlight to you is we've also got this really exciting project going around CSF, where we're starting at 4 of our mental health memory clinic sites and looking at the feasibility of starting to collect CSF at those sites. And again, for me, that is about expanding access of this research to real-world patients, as well as the hopeful benefit of them being able to access biomarkers. So James Rowe talked to you very briefly about the Readout project yesterday. I'm just going to explain a little bit more about what that involves. This is a project funded by ARUK, the Alzheimer's Society, NIHR, and also the Postcode Lottery. It's quite an ambitious project. We've designed it very much with patient and public involvement in mind, but we will be recruiting across our network of 52 sites. What we are consciously going to do, what this project is basically going to look at is what's the utility of a group of biomarkers for diagnosing or detecting prognosis, both in Alzheimer's dementia and non-Alzheimer's dementias. But the thing that we're doing, maybe that's slightly different from other projects to date, is that we are going to recruit 30% of our sample from underrepresented groups. So 20% from ethnically diverse group with a strong focus on the Black and South Asian populations in the UK, and 10% of people with comorbidities. And our exclusion criteria are basically nil. So if people are very elderly, if they have complex medical histories, we still want to be able to include them. And then we're going to include 10% of people that we've called not engaged. So these are people who we think probably should be coming to a memory clinic, but they're not at the moment. Mindful of the fact that over the next few years, if we do have disease-modifying drugs licensed, the people coming to memory clinics are likely to be shifting over the next 5 years or so. So we need to be prepared for that. And we're working really closely with the Global Alzheimer's Platform. I'm really pleased to say Lynne Hughes is talking with us this afternoon. And we're going to utilize some of the processes they've used very effectively in the US to increase recruitment from these diverse populations. So using things like research buses to be able to do these accessible blood tests at sites that people can actually get to. And I'm not going to go into this in detail. Unfortunately, I don't have time. If anybody wants to know more, please let me know. But broadly speaking, this study will have 2 arms. We'll have an observational arm for the first 3 years, where we are collecting data around a range of biomarkers, broadly classified into 3 tiers. James touched on this yesterday. So biomarkers where there's already good, robust data, already in clinical use in some countries. We've got an exploratory arm where we're going to be working with industry partners to look at some other more novel biomarkers, and we're also going to be looking at certain key issues around processing, like processing delays, or can we actually do this through postal tests? So there'll be 3,000 people recruited to this study. We'll hopefully get data around the cutoffs for these biomarkers, and by the end of the study, we'll understand what's the best panel or individual biomarkers we need to use. And then we'll go on and do a randomized controlled trial for 2 years, where we will look at the impact of clinicians and patients receiving their biomarker status on quality of life, carer quality of life, other symptoms like anxiety and depression, and also healthcare use. Because the other part of this study that's really important is what's the health economic impact of doing these blood biomarkers at scale. What this model will do, and I'm really pleased to say that we've got 14 integrated care boards. These are the commissioners that fund and organise services across England, already partnering with us. So they will help us design this proposal and this model. But what we really need to know is what value do these blood biomarkers actually give us at scale? And if you try and now extrapolate who's likely to need them in the NHS over the next 5 years, can we actually afford them? I mean, I sit very frequently in memory clinics and see patients where I just think I just need a biomarker. All I have at this point is often a CT scan, and I know David raised the point that, yes, I'm still doing the MMSE and the MOCA, which was what I was doing 30 years ago. So we haven't really moved on, and I need more information now. So my sense is that these will be affordable, they will give us value, but we need to have the science to understand if that's the case in real-world settings. And so hopefully at the end of this won't just be another piece of research where, okay, we publish papers and we're all very pleased that we've got great results, but we're actually working with the people who set up and pay for the NHS services so we can roll this out at scale in real-life clinical services if we can prove that they're useful. So for the next 12 months, and this is for the TDF and within DPUK, but also the wider infrastructure, I think we do need to become faster and more efficient. We need to be using studies like Readout, and there is another similar study funded at UCL called ADAPT that's looking at Alzheimer's dementia specifically. I think there will be a wider use of registries such as ours to characterize people ahead of trials. And I think we need to think about how we use these biomarkers and these cohorts to identify people in what I would call non-traditional settings. So we really need to make an effort to go out and enrich our sampling and to think, how can we leverage this wonderful resource that we have in the NHS, all this great lead-in data we have, but at the moment we really struggle to be able to join that up with a lot of the research data. So how do we make that work? And that's definitely one of our plans for the TDF over the next 12 months. And then finally, how can we build relationships and partnerships to understand the utility of these new biomarkers beyond the UK? And how can we deliver trials more effectively beyond the UK? So I'm going to finish talking at that point and introduce our first speaker. We've got a great range, as I said, of talks this afternoon. Hilary Evans, hopefully. You're with us, Hilary? Yes, come down. So Hilary Evans really doesn't need any introduction, but I will try. She is the Chief Executive of Alzheimer's Research UK, which is the UK's leading dementia research charity, but also co-chair of the Barbara Windsor Dementia Mission. And she's going to talk to us about progression. I think the UK goes large is the title of your talk. So look forward to hearing more. Thank you, Hilary.
Really lovely to be here with you all. As I've been introduced, I'm Chief Executive of Alzheimer's Research UK and co-chair of the Dame Barbara Windsor Dementia Mission. So I am going to be speaking wearing both hats today. I hope you'll be pleased. I don't have any slides, and I won't be speaking for too long, but want to cover a number of different areas which I think are really exciting. But firstly, just want to start by congratulating DPUK and John Gallacher for your leadership in convening what is a fantastic conference and bringing this community together to discuss the really important and exciting developments in dementia research. So thank you for inviting me to speak. As Vanessa said, the title of this session is The UK Goes Large. So I'm going to use that prompt to talk about where we are, the big picture, the big ambitions that I have for people with dementia now and in the future, and the big opportunity that's in the UK when it comes to clinical trials. So if nothing changes, 1 in 2 of us will be affected by dementia, either by caring for someone with the condition, developing it ourselves, or both. So that is the stark reality that we face, and I'm sure many of us can relate to this in our own families as well as our professional lives. I have a deep connection to this cause. 3 of my grandparents died from dementia. And in fact, 2 of my 2 grandfathers spent many hours in this very building as trade union leaders. So it's fitting that I'm here standing up for those who don't always have a voice in the way that they did many, many years ago. I also have one aunt living with dementia and one who's recently died with Alzheimer's disease. So this is a very personal and professional mission for me, like many of you in this room, I'm sure. So that drive to change the reality of dementia, to make it become something of the past, not the future, underpins everything that I do. So I think the good news is that we are in probably the strongest position that we have been yet to end the devastation of dementia. We've seen tremendous breakthroughs which have the potential to transform how we view and respond to diseases like Alzheimer's. The power of research, building on decades of scientific endeavour, means that we're now at a tipping point for progress in dementia, with the first disease-modifying treatments on the horizon, as well as game-changing developments in biomarkers, as we've heard, and the powerful potential of big data to really accelerate that progress. The arrival of lecanemab and donanemab, the first Alzheimer's drugs that will tackle the root cause of the disease rather than just the symptoms, proves what we've always known, that through research we can find a way and hopefully one day cure the diseases that cause dementia. But this will just be the tip of the iceberg. There are now over 140 Alzheimer's treatments being tested in trials, so it really is a matter of when, not if. New drugs are being developed that prove to be even safer and more effective. I want to focus my remarks today on an area which is vital for the future of this agenda, clinical trials. We all know that this is an essential part of medical research, providing us with robust evidence that we need to assess the promising safety and effectiveness of new therapies. Success begets success. Approved treatments will mean a growing pipeline in dementia, which will bring great opportunities for further investment in dementia research. We now have that unique opportunity to position the UK at the forefront of this revolution, driving economic growth while enabling patients access to promising new treatments. If we are to achieve this, we need to ensure that our clinical trials infrastructure is ready. Over the last 5 years, we've seen a concerning decline in both the numbers of trials being initiated in the UK and the number of patients recruited into these trials across disease areas. COVID, of course, has played a part in this, but the UK has been slower to recover than other countries. This is compounded by particular challenges for dementia research, particularly in recruiting participants to trials. This starts with a lack of clear diagnosis in the early stages of the disease and variable access to diagnostic tests. Recruitment of patients for research is also often not a priority in clinics that are dealing with huge backlogs at the moment and increasing demands for care. The lengthy study setup times and bureaucratic negotiations around contracts and costings mean that many people who will have been screened and are ready to take part in research become ineligible by the time the study can start. So although there are nearly 1 million people living with dementia in the UK, only 2% of those with the diagnosis are currently registered to hear about dementia clinical trials, with only a proportion of those ultimately taking part. It is estimated that the UK participants make up just 2.5% of the total phase 3 clinical trial participants in dementia worldwide. In 21-22, just 61 participants were recruited to late-stage dementia clinical trials supported by the NHR Clinical Research Network. This was 10 times fewer than trials in stroke, heart disease, and 100 times fewer than cancer drug trials. People living with dementia in the UK are not offered the opportunities to take part in research and clinical trials, both in comparison to other health conditions and other countries. And those who are most likely to develop dementia are often unrepresented in trials. For example, although Black people are twice as likely to develop dementia, 94% of participants in Alzheimer's studies are white. In addition to holding back progress, this deprives people of the benefits of taking part in research, including access to medicines that they would not otherwise receive and the opportunity to make a valuable contribution to the development of treatments that will change the outlook for people in the future. This is really key to giving people hope. There has been work in the UK to address these issues. We've seen a government review led by Lord O'Shaughnessy setting out ambitious recommendations for change, but we need to go further. To address some of these issues, our team at Alzheimer's Research UK set out a vision of how we can do this in the UK, and we have 5 principles. The first is speed. Enabling faster study setup through streamlining ethical approval and contracting, as well as through innovative trial design and delivery. Second is the establishment of a minimum clinical data set shared across trial sites so that patients can be moved into screening quickly and efficiently. Thirdly, a central hub to connect sites and offer support to the best practice sharing, including around diverse populations. Fourthly, ensuring any network of trial centres includes a range of sites to build capacity across the system and across the UK, not just in the Golden Triangle leading research areas. Our 5th area is around alignment, and that's with other initiatives in the UK and globally. The final point here, I think, is a particular strength that we have in the UK. We have key initiatives in this space to build more ambitious, joined-up approaches. And DPUK, it really is leading in this space with its trials delivery framework under the fantastic leadership of Vanessa, as we've just heard, and John. A strategic and collaborative approach is crucial for enabling progress. It's what we saw with COVID, with the vaccine task force. So with this in mind, Alzheimer's Research UK advocated for and strongly welcomed the establishment of the Dame Barbara Windsor Dementia Mission. This aims to fast-track the development and delivery of new medicines for the diseases that cause dementia, bringing together researchers, industry, government, the NHS, regulators, and people affected by dementia, alongside key initiatives that we already have in place, such as DPUK. We were delighted to receive the support of the government in setting up this initiative, and last year I was honoured to be appointed as co-chair of the mission alongside Nadeem Sawar. The Dementia Mission seeks to take an ambitious and joined-up approach, building on the talent that we have in the UK to maximise our impact. It also aims to position the UK as a key leader in dementia research globally. This will boost our economy, as we know that dementia research is a leading growth area. Recent modelling that was commissioned by Alzheimer's Research UK has showed that for every £1 invested in dementia research, it's generating £2.59 within the UK economy. So fantastic return on that investment. The Dementia Mission itself will be focusing on 3 key pillars. Firstly, identifying, validating and implementing a suite of decision-enabling biomarkers. This is essentially what had been referred to, I think, in recent years as the Neurodegeneration Initiative, or the NI. Enabling faster and more innovative dementia clinical trials and ensuring the implementation of new therapies in the NHS through aligning translational research, clinical practice and regulatory frameworks so that people in the UK are among the first to receive dementia treatments. The mission itself is ambitious, and in the last 12 months, we've really broadened out the scope and the funding of the mission. I'm delighted that the new Dementia Translational Clinical Trials Network has been established by NHR's Dementia Translational Collaboration under the brilliant leadership of Kath Mummery, and obviously is working very closely with the Dementia Mission. This aims to build capacity and expertise in the early phase dementia trials across the UK, widening access to participation through a unified network of dementia trial sites across the country. Its initial focus was on developing a streamlined set of regulatory processes to speed up the delivery of trials and the infrastructure needed to provide a centralised support and information, as well as integration with local NHS services and communities. In addition to, we are working closely with the Trials Network. A Dementia Trials Accelerator was announced in the government's autumn statement last year. So this was an extra £20 million of government funding that we put a business case forward for. So this will be delivered jointly by the Dementia Mission, but with the Medical Research Council. this Dementia Trials Accelerator aims to embed more innovation in how clinical trials are designed and delivered in order to increase both the speed and quality of large-scale trials. This will include a focus on enhancing diverse recruitment through optimizing existing data sets and digital tools. It will aim to build on significant achievements and work in collaboration with DPUK. The accelerator aims to capitalise on existing population cohorts, developments in biomarkers, and UK leadership in big data to help match people to studies, reduce the unacceptably high failure rates in trial participation, and I'm really looking forward to working with key partners in the development of this work. We are delivering the mission's ambitions through partnership with those who understand the challenges in developing and implementing new treatments and diagnostics, with industry, academia, the NHS, regulators, and importantly, people with lived experience. We aim to establish a public-private partnership this year to deliver the mission's objectives, to ensure that we can combine and build on resources already at our disposal, such as the fantastic research institutes here in the UK, because we need to tackle these challenges together. We know that a key part of this puzzle is people. We need a new generation of clinical researchers in place to drive a revolution in dementia clinical trials in the UK. Everyone in this room will appreciate the challenge for clinicians to be involved in research, not least when the NHS is under considerable pressure. As a charitable funder, Alzheimer's Research UK must play its role in helping to address this. That's why we're currently developing our clinical research programme to bring more clinical trials to UK for people living with dementia. This will consist of grant funding calls to increase the number of research active clinicians and bolt-on studies to existing clinical trials to help validate new measures for use in dementia, as well as collaborations and partnerships to build on existing infrastructure and initiatives. With this programme, we want to ensure the highest level of innovation and maximise the value from every clinical trial. As a result of these initiatives and working with partners such as DPUK, I feel very hopeful about the future of dementia trial delivery. Last month, as we've just been referenced, I was delighted to attend a reception at 10 Downing Street, which brought together charities, researchers, investors, industry, NHS leaders, and people affected by dementia to celebrate progress being made in dementia research and to discuss some of the next steps of making the UK a global leader in the field. At this event, it coincided with the appointment of Scott Mitchell, husband to the late Dame Barbara Windsor, who died of dementia in 2020, as People's Champion for the Dame Barbara Windsor Dementia Mission. Scott is a passionate ambassador, an advocate for dementia research, so his role really will help in delivering that lived experience of people affected by dementia and making sure that they are at the forefront of everything that we're doing in the mission. Experiences such as Melissa Williams, who shared her moving account of how dementia has and continues to impact her own family. As Melissa said at this number 10 event, a cure cannot come soon enough. Melissa's personal account was tremendously inspiring for a number of us in that room. And as I said at the start, it's what drives me too. We know from other conditions like HIV and AIDS that the first generation of treatments had limited effectiveness, difficult side effects. But these paved the way for combination medicines that revolutionised outcomes for the next generation of people with the condition. And like HIV and cancer, much of the progress in dementia was held back through fear, lack of understanding, stigma, until those treatments started to come through. Faster, more innovative clinical trials are really going to drive this progress. Over the next 5 to 10 years, there will be a transformation and we will see more treatments come through, targeting different processes and being used in combination. And we'll have the tools to detect and tackle diseases at a much earlier stage. We should celebrate this change and what that means for the many lives affected by dementia. But these breakthroughs bring new challenges. We must take inspiration from the developments and redouble our efforts to bring about more effective drugs. It won't happen overnight. It won't be easy, but along the way, lives will be changed, and one day, lives will be saved. We will move closer to my ultimate hope of a cure. For me, this is the first step in that journey. The outlook for dementia and its impact on individuals, family, and society is finally changing. I look forward to continuing to work with the people in this room and the wider dementia research community to fulfill all of our ambitions for the dementia mission and to drive forward the development of much needed new treatments towards the cure that we all stand for at Alzheimer's Research UK. Thank you.
Thanks, Hilary, for your visionary statement. Philip Bath from Nottingham. I was concerned to hear your very low recruitment rate. And although we in the UK are very fortunate to have the world-leading clinical research network, obviously evolving into the RDN, it has had one problem which was unintended, which is this silos of dementia and, in my case, stroke and so on. We actually were recruiting in the year you quoted, and probably recruited that many patients into people who'd had a stroke, but where the primary outcome was related to cognition and dementia. And of course, the way of counting precludes, or the way back then of counting precludes really looking at what's going on across the UK, not just in dementia named trials. I'm hoping the RDN is going to change, and I've been in contact with them. And it looks like they're going to stop this accounting by the originating silo and start to sort of look not just at the input, but also the output, in this case, dementia. So cross fingers, actually the numbers will look better than they really are, than they appear to be.
Thank you.
Yeah, really valid point, and actually good to get those updates, more than I've heard so far from the RDN, but that sounds very promising. Any other questions for Hilary at this point? We will be having a panel discussion at the end. I think you're here with us for that. But anything else at this point?
Yeah, I think just in response to that point, if there aren't any other questions, I think that we do need to ensure that we're taking a really holistic view here. And we do, you know, the way that we are going to be thinking about the diseases that cause dementia and how they are then thought about in terms of bringing people forward for trials and what are the other kind of comorbidities. There's lots of things that we need to ensure that we are not just sort of counting in a very kind of siloed way.
Okay, well, once more, thank you very much, Hilary. Really lovely to hear from you.
This isn't DPUK, this is a newborn baby. We just established or incorporated a non-profit foundation in the Netherlands about 6 weeks ago. Just opened a bank account yesterday, so we're open to accept donations. It's a new initiative, so what I can share with you today is kind of the vision and the general strategy of InRAD, the International Registry for Alzheimer's Disease and Other Dementias. But let's start with the main questions. What is actually driving this project, and why do we need another registry? So unfortunately, the most recent publication kind of on the landscape of global registries is from 2017. Obviously, there's a couple of new. ALZNet is maybe one that I should mention here, the US Alzheimer's Association's new real-world registry. But in principle, or in essence, I think what's shown here is still true. So there's a couple of amazing efforts. There are registries such as SWEDEM, the Swedish Dementia Registry, which have been around for decades, have enrolled tens of thousands of individuals. Most of these registries are not practice-based and are not really designed to collect real-world data. They're designed to answer research questions, to do epidemiological studies. They don't offer necessarily a value at the point of care for the clinicians. So what we need is a different type of registry, which is international and which is practice-based. So the kind of general vision for InRAD is to form something, to build something which is a catalyst for change and practice-based Alzheimer's disease research, which is important because we're just entering a new era of Alzheimer's disease treatments. And also the kind of the landscape of stakeholders is developing and shifting, and there are regulators, for example, there are payers. They all have their different needs. There's not only us clinicians and academics, and we have to address those needs and build something which is able to provide the data to do analyses, to answer questions which are relevant for regulators and payers, for example. there's a lack of standardized data across all those amazing registries. There is no agreement on a minimum data set that needs to be collected. I'm just trying to, I will just show you in a couple of minutes how we approach this question. And I will also kind of try to outline some of the challenges that we're facing in agreeing on something simple as the main cognitive outcome. And I was very naive moving into this kind of consensus process, and I've learned that it's quite difficult even to agree on something as simple as using the MMSE or the MOCA or something else as your main cognitive outcome measure. Even that is almost impossible. There are all these different national registries. They need to be somehow aligned. I don't want to use the word harmonized, because harmonizing is not good enough and it takes too long. We need some strategy to collect data that can be used for joint analyses. And our plan is not to replace any of the registries. We want to kind of create a thin layer that is somehow on top of the existing registries that provides a platform for collaboration and joint data collection. So the opportunities are quite big, I think. So we've talked to the European Medicines Agency, for example. We had a kind of an informal meeting but with all the main stakeholders from the large European countries, from EMA. They were quite interested in this effort, and they agreed. They didn't really want to share any of their trade secrets. We didn't really get any feedback what kind of data. Probably they don't even know what kind of data they will be wanting to collect. But there was a huge interest from EMA, and definitely kind of a hope that something like this registry would be available in Europe to collect real-world data. As I mentioned before, an agreement on a minimum data set is critical, I think. It will help us to kind of manage patients, provide information back to the clinicians at the point of care. We can, of course, use it for research, for epidemiological studies. And one thing that is obviously missing from all the ongoing efforts is safety data, because until now it didn't make any sense to collect safety data. There were no disease-modifying drugs. That's going to change quite soon, hopefully, in the EU and also the UK. We need something that's able to collect safety data as well. We also need to create something that allows the members of this network and also external collaborators to share data and things like data security, GDPR. I live in Germany. It's almost impossible to do anything with data. It's much better in the UK, I think. But there are huge challenges when we talk about something that's international. So what's the overall platform concept? And this is something, if it lets me, this is something that we developed in collaboration with MS Space. MS Space is possibly the largest, or one of the largest, multiple sclerosis real-world data registry. It's been around for 25 years, I believe, with centers all around the globe, in Europe, Asian countries, Saudi Arabia, Australia, US, Canada. And they've developed all the guidelines and governance frameworks, ethical legal frameworks that we are now kind of using and building on to develop our Alzheimer's disease registry, because it's a great template. And the idea is to have this platform custodian, which is now the Dutch non-profit foundation, which will manage and host the registry. It's a multi-stakeholder collaboration, because there are partners from the pharmaceutical industry. There are main sponsors currently. Hopefully that's going to change quite soon. And then there's a group of experts, which we call, and which MS Space calls an SLG, a scientific leadership group. So they're responsible for the content of the registry. So MS Space, for example, they have created MG Base now, Myasthenia Gravis, registry, which uses the same frameworks, and there's a separate scientific leadership group for MG, because the MS experts don't have the specific expertise required. So for Alzheimer's disease, we will we are in the process of building this Alzheimer's disease SLG. There will be independent statistical advice, and then there will be members of the network in all the countries that want to collaborate. And the idea is that we have this common kind of minimum core data set, which everyone is expected to collect. That's going to be quite lean. And then centers can decide if they want to collect additional data for sub-studies. And the system will be flexible enough that it's quite easy to just talk to another center, decide you want to do something on a specific topic, you add a few fields in your registry platform, and you start collecting data that you need for your sub-study. Or if a drug is approved in a country and there are country-specific requirements, there will be an option to add additional fields to collect data that the regulators in, let's say, Germany require if lecanemab or donanemab are approved. So it will be adaptable, flexible for all the different needs and provide a platform for collaboration. And those are the advantages for the members, and we are actively reaching out to sites now. So if anyone is interested in joining as a pilot site. The most important point probably is data sharing and data ownership. And the model that we are copying or have copied from MS Base is that data control, data ownership always remains with the participating sites. So as a site, you never give up data control, data ownership. It's your data. You put it on the platform, which then enables other members of the site or external collaborators to work with the data. And MS Base has implemented actually quite an attractive and effective collaboration model, which basically requires sites to opt out if they don't like a research question. So if a member of InRAD will want to answer a research question, an email goes out pretty much to all the member sites, and they have 2 to 4 weeks to opt out. If they don't opt out, their data can be used for the analysis, which I think is a very nice approach. It enables research, and it still allows all the participating sites to retain ownership of the data. And the foundation, the InRAD Foundation, is only the data processor, never the data owner or controller. That's just an example from MS Base. It will look similar in InRAD. That's the that's a clinician dashboard. It's actually the information that you see at the point of care. So if you see a patient in your clinic, you open the InRAD registry. You are required to enter a few basic data points, and you can see all the visits over time of your patients, all the medication, comorbidities, if they're on a drug, adverse events, which I think adds, at least from what I've heard from MS Base, quite a lot of value for the clinicians. They're not going to get paid, so there will be no visit fees. This is not a prospective new study that's fully funded, so we need to provide clinical value and something that the clinicians perceive as something which not only adds burden because they have to enter additional data points, but which they can use to improve patient care in their clinics. Just briefly, the timeline. So we had a kickoff meeting last July at the AIC meeting in Amsterdam, which attracted quite a lot of interest from clinician academics, industry, about 60 people in a room which was much too small. There was phase 1. We're just kind of finishing phase 2 now, which is agreeing on the minimum data set that we want to collect and publishing this data set so that it can be used by any registry that's interested in collecting kind of this harmonized consensus-based data set. And then hopefully we'll be able to develop the kind of the full platform until later next year and roll out the full registry sometime within, let's say, 18 months or so. There's a website if you want more information. There's also a LinkedIn page, which I had to learn how to use, and the project team sometimes forces me to post, which I'm not really that experienced in doing, but you can have a look at the LinkedIn page. And just to briefly showcase how the minimum data set is agreed on and how we'll use that. So the idea is there are different national registries. Our registry is one of them. And then there's this kind of consensus-based data set that we want to collect and that other registries are also allowed to collect if they want to participate. And then there will be some registries which don't want to collect this data set. But the data set pretty much enables data collection, harmonized data collection across different registries globally is actually what we're trying to achieve. So we did this kind of a modified Delphi consensus. We didn't do a full Delphi because we want to do it relatively quickly. And it's based on a consensus paper that was published last year by a group chaired by Giovanni Frisoni. I'm going to show you kind of the high-level results of this Delphi consensus on key outcome measures that are relevant for Alzheimer's disease patients in clinical situations. So we built a steering committee, which is very international. Kath Mummery represents the UK. We have colleagues from Japan, Australia, South Korea, several European countries. We wanted to get a broad consensus with input from different regions of the world. We had feedback from the European Medicines Agency, as I said before. And then we also had a broader stakeholder consensus, which included partners from pharmaceutical industry, NGOs and clinicians and academics. And we're just in the process of consolidating all the feedback and then agreeing on this minimum data set at a final steering committee meeting in May, and then publish the results. So just kind of high-level outcome measures that were resulted from this published Delphi consensus. So the domains they looked at were cognitive abilities, functional abilities, behavioral and neuropsychiatric symptoms, quality of life of the patient and the caregiver, health, social care and treatment-related outcomes, medical investigations, significant disease-related events, and also global outcome measures. And that's kind of, those are the numbers, how many different assessment instrument and outcome measures they screened. And then just as an example for the cognitive domain, that included the MoCA, the MMSE, ADAS-Cog, the Ray auditory verbal learning test, the train-making test A and B, and the Edinburgh's revised battery. And that's kind of the short list of cognitive measures that we also added, or that we also discussed in our modified Delphi consensus, and with the same thing with all the other domains of interest, such as functional abilities, for example. So the respondents, just to be fully transparent, were these 4 pharmaceutical companies. So that's feedback not from one individual, but we asked, let's say, Eli Lilly, please go back to your teams and let us know what your feedback as Eli Lilly is, and with the same thing for Eisai, Biogen, Roche, and Novo Nordisk. We had feedback from the Alzheimer's Association in the US, Alzheimer's International, Alzheimer Europe, and also Gates Ventures. And we had feedback from 72 clinical academics from all the different countries that you can see in the list. So it's actually quite international. And one thing that makes me a little bit sad is that most of the clinicians were neurologists and only a handful of them were psychiatrists. This pretty much represents probably the global landscape of clinicians that provide Alzheimer's disease care. I would like to see more psychiatrists outside of the UK also being interested in this topic. And then just very briefly, the results. So what we did is, like in a Delphi, so if 70% of the respondents agreed that variables should be selected either as part of the minimum data set or as part of a more extended data set, then we kind of, that counted as agreement and a successful consensus. If it was somewhere between 60 and 70%, we had a discussion. It was below 60%. That was definitely a variable that we're not going to collect in the minimum data set. And there are very obvious things like patient IDs and if the patient has provided consent or not. There are things which are a bit tricky, which is ethnicity and the kind of coding that we now agreed on to use doesn't make any sense for me as a German because it includes ethnicities which I've never seen in Germany. And I have a very biased view because that's kind of a difficulty or a challenge in kind of in terms of research in general in my country is that ethnicity is not a very big topic. That's much better in the UK. So there were difficult questions which needed a lot of discussion. We decided to have all the lifestyle factors, which are very important as part of the extended data set that a site is not required to collect, but which the site may decide to collect. And the logic behind all of this is basically, this is practice-based. It's not a fully funded prospective new clinical trial, and it needs to be lean, and the burden that we add to the busy clinicians' life has to be as minimal as possible. So that's why we decided to concentrate on the most important outcome measures as part of the minimum data set. Everything else is extended. And then there's the big question that I briefly touched upon earlier is what's the best cognitive outcome measure? And I thought naively that it would be quite obvious that the MMSE or the MoCA would end up in the minimum data set. That's also what the US AltsNet are using. So you can use the MMSE or the MoCA depending on disease stage. But there's still an ongoing discussion in the steering committee now. Do we want to actually mandate MoCA or MMSE? Or would it be enough to just mandate some kind of clinical staging, such as the NIAA stages 1 to 6, so that you can do whatever cognitive test you want to do, whatever you do in the clinical practice, and use that to provide information on the stage your patient is in. And then everything else is quite clearly extended. So potentially the clinical dementia rating, some of the boxes, will be mandated by certain countries or regulators in certain countries. Then that will be added, obviously, as a requirement. But other than that, no one wants to do a CDR in clinical routine. It doesn't make sense to put it into the minimum data set. What I find, that's probably the most important information, quality of life. That's why I wanted to show it. But again, we don't usually collect information on quality of life. That's why it can't be in the minimum data set, but we will do whatever we can to collect patient-related outcome measures. We're looking into technological solutions to use solutions where the clinician doesn't have to enter any data, but it's collected, kind of it's a home-based assessment or in an app, something which the caregiver, for example, can complete before the patient actually shows up in the clinic. But we definitely want to collect these quality of life related outcome measures. And then Vanessa asked me to show this slide. I have no idea what it shows. You mentioned it briefly before. I can't repeat it. What I'm trying to say here is, so I am, as a representative of InRAD, I'm very interested in collaborating with DPUK. And we should definitely try to explore opportunities to align our data collection. That's what I'm trying to say here. Thanks very much.
Great to hear that, Robert. So I guess the one thing that I did want to raise, so in sort of, it sounds like you had your cutoff in 2017 in terms of sort of initiatives. I'd very much like to point out that since 2019 at DPUK, we have set up the largest clinical studies register where we have over 68,000 people with genotype and phenotype. Together with Sarah Baumeister's team, we've worked on the problem that you're dealing with now in terms of what is the standardized data set, and we've come up with some variables. We are doing remote genotyping for these people, now we bring them into blood and so on. So I think what you were saying about learning from each other, I think we definitely have, something we can share, so we'd be very happy to talk to you.
Thanks very much. I couldn't agree more.
Thanks, Robert. A quick question. In large trials, and my background is stroke, where we do this a lot, we use telephone follow-ups. So we can do very simple, low-cost follow-up in large numbers of people. We actually record telephone mocker and or MMSE. How do you, could you extend your data to allow telephone follow-up? Because it really is so much a better way to get really big numbers.
Yeah, I think follow-ups is probably the most important topic that I haven't mentioned, because in some of these very large registries, dementia registries, there's almost no follow-up data. They have 100,000 individuals enrolled over 20 years, and they maybe have one or less, on average, less than one, some of those very large registries, less than one follow-up visit for the 100,000 individuals enrolled, which slightly reduces the value of the data collection. We are looking into technology solutions to collect cognitive outcome measures, for example, regularly. I think the first version of the registry, which is challenging enough to build, will be supporting clinicians in doing whatever they do in their clinic. And there will be no, let's say, we'll try to get, it will be impossible probably to tell a clinician you have to see your patient every 12 months. So it's not a prospective study, it's real world. And at the start, at the beginning at least, it will be whatever the clinician collects, and hopefully they will agree to collect this minimum data set so that we have at least an MMSE or a MOCA or whatever our steering committee agrees on that should be collected.
Great. So unless there's any other questions, thank you again. Right, Robert.
Thank you.
So I just wanted to show my disclosures. As Vanessa mentioned, I was with Quintiles IQVIA for 23 years, with most of the time spent as VP and Global Head of Neurology. And those 125 trials were in the last 10 years I spent at IQVIA. So I did a lot more trials in Alzheimer's than the 125, and I have the metrics on all of those trials. But what I want to talk to you today about is the Global Alzheimer's Platform. GAP were put together. It's an organization that was founded in about 2014, and there were a number of pharma companies, 6 big pharma companies that put in the funding. They had 2 challenges to the GAP team. 1, be self-sufficient and self-funding within 5 years, which they did. And 2, look at ways to expedite drug development in Alzheimer's disease. And we've actually taken that into the Davos G20 collaborative for Alzheimer's. And GAP are not a CRO. They're not a pharma company. They're not an SMO. They're basically a high-performing site network in the US and in Europe. GAP put in concierge services to the sites. They do not look at data. They do not monitor data. They basically provide support for startup, and for GAP-sponsored studies, they've agreed with all the sites in the US, even the academic sites, that they can all use the same central IRB. They put in people to help with the IRB submissions. We know already what the imaging center is, where they are, what their technology is. We know their raters, we have their CVs. And because of that, startup for a GAP-sponsored study is around 35% quicker than a non-GAP study. The other thing they offer is something that Vanessa alluded to called the Inclusive Research Initiative. And that's a team of people who are not my color. They go into the communities in the US, into the African American communities, the Hispanics, the Latinos. They go in every month, whether there's a study there or not, and they get to know the community leaders, the church leaders, the hairdresser, the politicians. They start to build the relationship with that community. They give them educational leaflets, they do training sessions, they do memory health clinics and brain health clinics. And they gain the trust of the community. And at the moment, with the GAP-sponsored studies, 5 big ones in the US with big pharmas, the percentage of underrepresented populations in those studies is between 21 and 35%, which is quite amazing. So why are we here? GAP were challenged to expedite drug development. So we've piloted a couple of our own studies. We're working on about 17 other studies with big pharma, biotech, small pharma. But why do we have to expedite it? This slide is a lot of data. You can see the global clinical trial enrollment rate, and I hear the gentleman at the back. We are very low in Alzheimer's disease globally. 0.3 patients per site per month. That's on average 4 patients per site per year. And everyone will say, no, that's not right. Don't forget, 20 to 30% of sites never recruit a patient. That affects the statistics. We're not much better than some rare disease recruitment. And if you look at the time to start up in CNS, it takes us a long time, mostly with the imaging and the contracting. And this is a slide from our friend Jeff Cummings, and I think most of you will have heard of or know Jeff. And actually the data on this slide are absolutely shocking. He presented it a year ago at ADPD. And the green bar is the time it takes to recruit, and the blue bar is the duration of the study. So you can see for all the trials in Alzheimer's, for example, DMT, biologics, vaccines, MOABs, antisense oligonucleotides, takes us 3 years to recruit for a trial with a duration of 18 months to 24 months. And it's absolutely dire for neuropsychiatric symptoms. 4 years to recruit to a trial that takes 3 to 6 months for duration of the treatment. So we have to change this. This is a busy slide. I'm not going to go through it in much detail. Just pulled data on more of the challenges with Alzheimer's. Our trials take a long time compared to some other therapeutic arenas like cardiovascular and oncology. And I've broken it down into where those timings are coming from. Trial recruitment takes a long time. And the problem is aided and abetted by our high screen failure rates and our low recruitment rate. I ran a study in preclinical Alzheimer's for major pharma. The cost of our screen failures alone exceeded $1 billion. Just for screen failures, not for any other costs, including CRO costs. And for some of those big trials out there, Emerge, Engage, Envision, Clarity, the cost per patient, excluding screen failure cost, is around $250,000 per patient when all the costs are put in. So this is a really hugely expensive endeavor for companies, compounded by our challenges with screen failures and a low recruitment rate. I didn't know whether to show this one or not. It just shows the time from site selection to contract execution in the UK compared to other countries, and the time from ethics committee submission to approval in the UK compared to other countries. So we have a challenge, which we're working on with Vanessa, with Hilary, to see what we can do to address this. It's seriously concerning, and it puts us on the back seat when we're trying to engage with major pharma to bring trials to our country. So the 2 studies we piloted, Biohermes and Ophelia, named after Greek gods. Hermes was the son of Zeus, and he's the god of loads of things, including speed. Ophelia is the goddess of the personification of ease and simplicity. So that tells you a bit about both studies. One's quick and one's easy. So the Biohermes study, and some of you will have heard about it. It's a non-drug study. We started it in 2021, and Robin, who was talking about MRIs and imaging before, did allude to this study. We recruited 1,000 subjects in 18 months at 17 US sites. There were 3 cohorts, MCI, cognitively normal, and mild subjects. It was quite quick. It was a cross-sectional study, which is one of the downsides. But we challenged ourselves to overall have an underrepresented population in the study of more than 22%. We came up with 24% at the end of the study, and the breakdown is shown here. 19% in the cognitively normal group, 25% MCI group, and much higher in the mild group. So what did we do? Basically, it was a very quick study. We took family history. Everything on the left-hand side is what you normally do. So we looked at race, ethnicity, years of education, con-meds, trial history. Everybody underwent an amyloid PET scan with IXICO as a central reader. But then we compared it to a number of blood-based biomarker tests. And at the moment, just added in the Alamar MAG array, it'll take us up to 160 blood-based biomarker tests in this study. We also had a range of digital tests. Every patient had full genotyping. We had proteomics, 2 sets of proteomics, some markers of neuroinflammation, and a lot of A-beta 40, 42, and tau. And as mentioned, 24% came from traditionally underrepresented populations. And these are some of the participants in the study. We were funded by the participants. It was less for the blood-based biomarker and the digital companies, and companies like Eisai, Biogen, Lilly. Even if they didn't participate, they wanted the data that this trial produced. We had 2 sets of proteomics, one with Alan Levy at Emory, mTherapro, and SomaLogic. And I'm just taking all the samples now to Metabolon for metabolomics, transcriptomics, and lipidomics. Those analyses have not yet been performed. We did take blood from every subject. So we had 1,000 subjects with at least 60 mils of blood, aliquoted out. So we have 60 blood samples per patient times 1,000. So we have a full set of 60,000 blood, serum, plasma samples, mixture that companies have taken and they've used to assay and test their own blood-based biomarker on. And you can see the other partners there from Merck, Quanterix, Roche, Gothenburg. We used oral analytics for speech assessment, CogniView as a digital test, and Line as the digital clock drawing and some other tests. We also had Retispect for retinal imaging in a group of subjects. As mentioned, IXICO with the central reader, Pentara with Suzanne Hendricks did the stats, and we have ATIA or GNS Healthcare who were doing the AI for us. So what are we doing with these data? All the biomarker partners have the data exclusively for 18 months. So they can do their assays, look at their sensitivity, specificity, all against the gold standard amyloid PET scan. Of the biomarker partners, 5 of them have submitted for FDA clearance, for 510K clearance, to get their product approved. Most of the patients in this study went into other therapeutic trials, not necessarily ones that GAP were running at those sites, but the trial that the investigator thought was the best and the most suitable for them. And that trial then received a fully screened, a fully characterized subject because we gave them those baseline data. And the last thing we're doing with the data, we've got the Scottish Challenge, led by Terry Quinn, Professor Terry Quinn, who's sitting at the back, and Professor Frank Gunmore. And we said to anybody, send us a proposal of how you can use these data. I should have said these data are exclusively for these people for 18 months. After 18 months, after database lock, all these data are free and available to anybody who wants them. but there is that period of exclusivity. But for the Scottish Challenge, if there is particularly an early clinical researcher that's got good ideas, that can provide a proposal that could maybe give us some insights and push the field forward. We've already had, I think, more than 30 proposals come in. We're looking at them. We will give those people access to these data now before the embargo is ended. So I think we're also still accepting proposals. Terry's nodding. So if anyone is interested, please do submit. Aphelia, ease and simplicity. So after we did Biohermes one, AbbVie came to see us and said, we're starting a SAD and a MAD study, phase 1. It's going to be difficult. Can you do a shortened version of Biohermes? Because although we've got a reasonable budget, it's going to cost us a lot of money. It's their MOAB, their harbour study. And the screen failure rates are going to be high, plus the fact not many subjects really want to go into a SAD or MAD study. It's more challenging. So we put together Aphelia, and it's a short study. It's very quick, 60 to 90 minutes. We started a couple of months before we initiate the site, and it's basically mini-mental, some demography, medical history, con-meds. If they pass that, if they appear suitable potentially for the trial, we gave them the C2N Prizegiving AD test. Linus and Altoida were exploratory markers. And we were contracted to screen 1,600 for a 50-patient study. So what happened? This is just one of the cohorts. And at the GAP sites, we put forward 173 pre-screened subjects. Interestingly, 112 then failed AbbVie's protocol for screening on the amyloid PET scan. So now we're using Prizegiving AD2, which includes tau 217, not just the ratio. We've had 22 randomizations, and there's a gap of around 40 subjects there. Some are still in screening, and some have gone on to other studies. So the ones that were qualified have all gone on to studies or about to go into study. And at the non-GAP sites, they screened 487 subjects, and they've randomized 16. And the difference is $3.5 million just on this cohort on screen failures that has been saved by doing this study. And this just basically summarizes it, that if you do a pre-screener, you're still paying for the pre-screener. It's about $500 for the basic 60, 90 minutes, and Prizegiving, as you know, is around $1,500. But even that is cheaper than a $20,000 full screen on a subject. So really, with Biohermes and Aphelia, which were pilot studies, we think that we can efficiently identify subjects, that we can get the diverse population because we take our mobile unit, called Thor, into the communities. These subjects are very willing to engage once they get to know our people. We have a very well-established, characterized, and diverse data set that we are making available to everybody in 18 months and others beforehand. And it does reduce significantly, particularly Aphelia, the burden on the site. This quick pre-screener doesn't have to be done by your most expensive resource. It is quick. You can put the screen failures on your own database and maybe put them into registries, other trials. It's very efficient. So what are we doing next? Well, Biohermes 2, of course, starting next month. It's going to be the same type of study. 1,000 subjects, the same 3 cohorts. We have 160 plus blood-based biomarkers. I have 10 to 15 digital tests, full genotyping, 2 to 3 proteomics, in fact, multiomics, lots of them. It's going to be a longitudinal trial this time. So every subject will undergo MRI scanning for Alzheimer's dementia and vascular dementia, and IXICO are managing the imaging for us. All subjects will have an amyloid PET scan, and all subjects will have a tau PET scan. We're asking people, as many as possible, to come back at 12 and 24 months to repeat the scanning, the blood draws, and the tests, and fully accept that a number of these subjects will be well into another study, and that's fine. We want to encourage subjects, if they're suitable, to go into a clinical trial, not to sit waiting on a database. We are looking to enroll 30% underrepresented populations, and we're bringing this study to Europe as well, working very closely with Vanessa and the Dementia Mission and Kath Mummery to put some sites in the UK and probably Spain and the Netherlands. The protocol will be finalised next week. The first patient is due in June. Again, all the data will be available to all the participants, the blood-based biomarker and the digital companies. We will merge the databases. We've also put our Ophelia data from the pre-screener available for everybody, and that's 1,600 subjects of data in the database. And patients can go into other trials immediately thereafter, and they're welcome to use the screening data and the images as their baseline data if the pharma company permits it. We're looking at working very closely with Vanessa and a major pharmaceutical company to see if we can implement a pilot study of a pre-screener in the UK with TDF sites. Can we do it? We won't have the pharma company's protocol now, but there are trials ongoing. So if we can show the pharma company that this is working, then it puts us in a good position to get their phase 3 studies that are coming along next year. At the moment on their list, we rank 27 out of 30 countries. They're taking the top 15. Our aim, working with Vanessa and the TDF and Dementia Mission, is to get us into that top 15 so that our people in the UK have the opportunity to participate in their next clinical development plans. And we're also working, as Vanessa alluded to, with her on the readout study to recruit underrepresented populations. So that's all I have. I didn't have many slides. I thank you all. And I know I'm between now and coffee, tea, or questions. So thank you very much for your attention.
I guess one question I would have, Lynne, is you've demonstrated really effectively the power of having a very simple pre-screening protocol. I mean, in terms of what the simplest could be or what we need now in a pre-screening protocol, bearing in mind what you've also highlighted around biomarkers, the utility of a plasma pre-screen versus, which one we use, do you have a suggestion of what we would need if we were going to roll out maybe an international pre-screening protocol?
That would be interesting. We are working quite a lot with our friends at Linus. Their digital clock is very quick. It's easy. You don't really need training to do it, so I'm told. So that would work. It's one that we would maybe with you put into a pre-screening protocol. Mini-mental, all the regulators use it. It's on all the labeling. None of us like it. But what can we do? We want something quick, cheap, to enrich our population so we're not putting people through a 20,000 screening process, which demoralizes the sites, the patients. It takes time. So Linus may be digital, and we're going to see what Feasibility turns up like. But I understand Fujiray Bio, who have liaised with LabCorp, have put their blood-based Tau 217 in for FDA approval. They've got the CSF 217 approved, and I understand the blood-based one is in there now. We don't know if they're going to bring it to Europe. talking to them. Everyone hopes they will, but that could be quite efficient as well. But we might get some others after Biohermes too, like the dry blood spot.
Yeah, which would certainly be, as you say, even more scalable. Thank you.
Thank you.
We have a question at the back.
Okay.
John.
So Lynne, first of all, congratulations. I think GAP deserves a lot of credit, and I know the work that you put into it. In DPUK, what we've found is that it's been very difficult to persuade industry partners to take risks. And when I look at the Biohermes, I'm thinking you have a shed load of people willing to take risks and to invest in that. How did you do it?
They want to do the study themselves, but they wanted the data. The big companies, the Biogens, Eisai, Lilly, Roche, they don't want to do these studies, but they want the data. And the biomarker companies, they believe their product's going to work. They believe it's the best. They have to validate it. And for a small investment in the study, they got a $15 million study and a potential filing. So, and it could be a lucrative market for them if their product works.
Yeah, okay, lovely. Thank you very much.
Thank you. We've got some more time, so a question at the front.
Thank you, Lynne. Wonderful talk, and looks like I mean, as the host said, that it looks like a very efficient platform for everyone. Is this platform also facilitating studies exploring those treatment for neuropsychiatry symptoms, so NPS, in late stage dementia, or only in those early stage and preclinical patients? And if so, if you do also facilitate studies in late stage dementia, following the previous question, what kind of test would you consider an efficient and effective test?
Blimey, I wish I was at coffee. No, we're not planning in later stage Alzheimer's. This was a pilot study. You know, ADDF, Gates, ADDI helped fund it. We had no clue what would happen. We're stunningly impressed on how well it worked. But we are we're working with MJF and a couple of others to see if we can do a Biohermes or some other name in Parkinson's, so that we can characterize with different biomarkers, and will be another year away. If the industry, if the field want another Biohermes in late stage Alzheimer's, I don't think we'd have a problem doing it. We would get the companies together, work out how to get the funding, which is what we've done. We'd pilot it, we'd use the same sites where we could, if that makes sense. I really want to make sure it's in Europe. And as for which test, I wouldn't know at this stage.
Thanks very much. Another question over here. We'll make that the last question then. It sounds like we all need coffee, so.
Yeah, no, thanks for the presentation, Lynne. It's super interesting. I think something that caught my attention is the fact that you have this community engagement, which is quite unique of the study. So I just wonder whether you've already seen, some of the results from those champions in these specific communities in those two studies, or whether you expect an increase of diversity in the studies as well.
Yes, I guess it's whether the different procedures you're using to engage diverse communities, does that increase recruitment go all the way through to the clinical trials, yes, as well as the kind of pre-screeners?
It's difficult to say. The ladies and chaps have been going into these communities for about 4 years, and we started this study 2.5 years ago. So they already had the relationship, and they've gotten to know a lot of the people. And once you get their confidence up, and also the neurologists and the site staff that we take into those communities, again, not my colour. They trust them. We don't have that issue in other places, but certainly I need to get Hispanics and Latino staff who can do rating scales, nurses, and where possible, if not a PI, a co-investigator to keep those people engaged. And they've been absolutely fantastic. 24% was our first attempt. The current study that we're doing is 35%. Retention is challenging. You have to be really engaged with them all the time. But recruitment, they want to go in it. And as Robin said, we're analysing the data in the different races and ethnicities, and there are differences. We're presenting it to AAIC and hoping that we can merge all our biohermes and aphelia data and have a more robust data set on imaging in the underrepresented populations that, again, will be available for everybody. Thank you.
Thank you, Lynne. Thanks again.
My name is Victoria Breeden. I work for Eli Lilly, and I'm really delighted to be here today with Dementias platform. Lilly has a really rich history with wanting to accelerate and work in the dementia area, so it's really nice to be here with you all. So thanks for having me. Part of my role is really advocating to keep research coming to the UK, and that is across all our therapeutic areas that we work within Lilly. So it is across the board, and some examples you'll see today is across all our therapeutic areas. So one of the things I wanted to kind of focus on is the fact that about a year ago today, the Lord O'Shaughnessy report was sanctioned. So he was asked by the government to create a report on the current state of clinical research in the UK. And it came with a lot of recommendations and government response. And there definitely have been some positive changes that we've seen in the past 1 year since that report was written. So the MHRA had a really bad year last year, I would say. They were very slow on their approvals for clinical research, clinical trial applications. We saw trials take over 150 days to be approved. So that was really having a bad impact, not only on the companies, but patients not being able to access trials, because trials at the end were just not even opened in the UK because of the massive delays. And within the last 6 months, things have really turned around, and across the industry, myself and my peers are seeing the target 6-day timeline being hit much more routinely. So this is really fantastic for us. And linked to that is some other items that were requested as part of the review, is that there is now a national contract value review that's been implemented. So the contract is a standard template. So as a sponsor, we can more effectively kind of have the reassurance of how long that negotiation period is going to be with contracting, because there's very little negotiation even able anymore. We have that standard wording, and linked to that is the interactive costing template. So again, when we want to negotiate fees with sites, there's very little that can be done. The lead site will do the negotiation, and the budget is then shared with the other sites of their information, and that has been working well for us. So there's positives, and even though the process is standardized, we are not really seeing a whole heap of increase in the speed to set up. So if we think about how at Lilly, what we are doing to assign trials globally is we're looking at speed to set up, speed to enroll, and meeting our patient recruitment targets. So that's something that we have to achieve. So I've highlighted speed to set up here because this is something that we're still seeing isn't really being met for the UK. In our large secondary care centres, we know there's a lot of complexity in the approval for the trials. There's R&D departments that have the overarching say. There's not only the research centre, but there's also the pharmacy, the scanning centres. There's lots of complexity at the site level, which we understand, but that is having a negative impact on our assignment of trials for the UK. And I've titled it versus the rest of the world because I think there's a thought that maybe we are only competing against some of the newer countries in research for Latin America. However, I have some examples here from some studies that have been approved recently and set up. So these are 3 studies from Lilly. And these are all the countries in each of the trials, and the date or the number of days is showing you how many days it took from regulatory submission to the site being declared enrolment ready. So you can see there's sites across the globe who are routinely faster than the UK in setup. So what can we do? We need to be making sure that all our everyone who's involved in the site for the research is aware much earlier on into the timelines so that we're not seeing these delays anymore. Another way of looking at it from, again, Lilly data is that a target we have is that when you open your first site in a country for your study, you want to be opening the rest of your sites within 6 weeks if possible to really capitalize on that enrollment period, to really maximize the number of patients that we are achieving in the UK. We have competitive enrollment in Lilly, so if your site is not achieving your patients, the patients will go to a different site. If as a country we're not achieving our targets, the patients will go and be found in a different country. So it's really important that we are fast. And you can see from this graphic, the target is 90%. France, Italy, and the UK are the ones that are really falling behind the curve. The light blue you can see are the ones that we have a potential to turn blue and become enrollment ready within our timelines, but we're not seeing that. They are going to the orange color, which are not achieving the enrollment readiness within that 6 weeks period. So we are losing out on patient enrollment in the UK. So what about speed to enroll? When are our sites engaging patients? So here are a couple of time points, which as a company we think could be considerations. So we have approval of your clinical trial application, approval and contract executed, approvals, contracts executed, confirmation of capacity and capability, site initiation visits, or actually wait all the way until the sponsor gives the green light. What we would love is for sites to be looking for their patients at that time we get the approval in the country. This is going to give us the most competitiveness as a country to make sure we're finding those patients as soon as possible. And we're not asking you to do protocol activities. We're asking for things like scanning medical records, making sure when you're having interactions with patients that research is actually discussed with those patients. In primary care, for example, they will set up research events when they know a clinical trial is about to open for them. And so they're inviting all their potential patients to come and hear more about that trial. So in primary care, they're having really great success. So it's something that we'd like to really see in secondary care as well, where possible. So back to the O'Shaughnessy report, another recommendation is to double recruitment into commercial clinical trials within 2 years, and then a further doubling by 2027. This was 1 year ago, so we're 1 year in, and with 1 year to go, it's just not fast enough for industry, and I can speak on behalf of other innovator companies. It is damaging the impact the UK by not recruiting fast enough into these trials. So I have another example, and this study, to be fair, was heavily delayed by the MHRA, and it was eventually approved in September 2023. And this graphic was taken in March this year. So we're showing the number of patients in treatment and the number of patients in screening. Why am I showing this? The UK isn't there. The UK had 0 patients, okay? So 5 months, 0 patients were enrolled into this trial. If I took another screenshot today, it would be 4. Just 100 to go in 18 months. Another recommendation from the report, which we really support, is UK-wide key performance indicators for trials. So pharma, we are measuring ourselves, we are measuring site performance, but we really think that site performance should be also measured externally as well. And something that we are concerned might be happening in some centres is that KPIs are being developed independently, and we see this as maybe holding up a contract execution, because as soon as that is signed, their internal timeline for finding the patient starts, but that's then delaying the opening for the sponsor, who could make the decision to pull that site from the study. So it's really important for us not to diversify here and make sure that key performance indicators are set up across the board for research sites across the UK. this is obviously really important, and it's workforce. And I know the NHS is super stretched, and having a sponsor organisation come and tell you you need to work harder in the NHS, that's not what I'm trying to do. I know we need more staff who are focused on research, and we have great sites who are full to capacity, and we know that because there's not enough staff who are going to be delivering the research. I think one thing that will help here is some transparency around funds. So when the sponsor is paying the sites for research, we know it goes into the bucket of the trust. However, I've seen there's a publication in the last couple of weeks where we need some more transparency around research funds. So I think if we can see where that money is going, it hopefully will encourage the trust to reinvest more money in those research sites or the research centres so that they can increase their workforce. So the UK is a great place to do research. I advocate for every day for more studies to come to the UK. We have a really wonderfully diverse population, and within Lilly, the UK is one of 7 countries who have been requested to support the US FDA required diversity targets for research. So that's a great opportunity. So we need to make sure we continue to support that. We're having those discussions with sites that set up to make sure that we are really having a diverse patient population, and we would really love it to reflect our census data. So that's what we are aiming for. And you might hear this, the word decentralized clinical trials, also accessibility attributes. So what are the different ways we can perform research without the typical, bricks and mortar centres? Within the UK, the opportunity here is that there's very little regulation against it. We are very flexible with how we can perform research. So we should be able to be really flexible and really innovative. So alternate sites for patient visits, electronic site files. And also a part of flexibility is the fact that we can, our clinical research associates can do remote monitoring in the UK. Lots of countries across Europe have more regulations and restrictions. This should be seen as a benefit for sites, as you have to host less people and hopefully spend less time on those monitoring activities with the source document reviews that are required by GCP. But we can do a lot of that remote in the UK. And we have so many patients eager to take part in research. I think since COVID, it's so much more well known. People know what a clinical trial is now. We saw, June Rain from the MHRA on the TV every evening, right? So, and also we've seen the NIHR be part of research app, and there's been a huge uptake in people registering for research. We just want to make sure the patients really do have access to that innovative medicines as soon as possible. So if we think a little bit about dementia, and I think this is a topic that has been covered really well by Hilary earlier anyway, the challenge is really for us in dementia research is the diagnostics. We don't have a standardised pathway for diagnostics in the UK. There's a lack of diagnostic capacity and infrastructure, which is preventing our people from accessing clinical trials in the country. So that's something that's really important for us to continue to work forward on through the dementia mission and other routes. So as clinicians, I think it's super important to put as many of your eligible patients through into clinical research as part of a normal clinical pathway for the patients. The clinical care through a trial can be so advantageous, the treatment the patients will have, and not even to mention the monetary gain from research for that site participating in the study. So yeah, ongoing work through the dementia mission. And that's it from me. Thank you.
Good afternoon, everyone. My name is Ta-Wei. I'm a psychiatrist trained in Taiwan and also a sleep medicine specialist. I'm currently doing my hopefully final year PhD in the IOP in King's College London. I think overall during the past 2 days, if you have been in dementia field for a while, you know that I think at the moment research are more or less focused on early prevention, early stage disease, and slowing the progression of dementia. But another very important and vulnerable population is those patients living with late stage dementia and also already developing some sort of behavior symptoms, so BPSD, behavior and psychological symptom of dementia. And what I'm going to talk about today is exactly focus on this group of population, talking about how bad they sleep and what we can know from a study with regard to the measurement for their sleep. So, I think although people are not so much interested in late-stage dementia patients, I think there are still a lot of research going on talking about sleep in dementia. I don't have to mention too much, but you probably know that sleep is one of the, it's linked to AD pathology, and based on some data published from the Rush study that Professor Bennett talked about early on, that there is actually a two-way relationship between circadian rhythm flattening, so sleep too much or not enough exercise in daytime, and progression of Alzheimer's disease and cognitive decline overall. And not to mention that sleep disturbances and nocturnal behavior symptoms are actually quite burdensome to their caregivers, and that is why many of the late-stage dementia patients, once they develop BPSD, especially nocturnal symptoms, they're sent to the care home, being institutionalized with poor quality of life. However, I would propose that I'm not going to only propose, but I'm going to try to convince you that the current status quo, the current measurement using questionnaires to measure these symptoms, the sleep symptoms, could be somehow questionable. So what I do for this study, for my PhD, is, actually I use a convenience sampling, recruiting late-stage dementia patients with existing BPSD, especially agitation and aggression, in a clinical trial called STAND. And I think I'm one of the victims of this COVID and MHRA bureaucracy that has been mentioned by Victoria and earlier by Lynne, for example, that although this trial STAND initially targeted to recruit 60 patients, but it is initially delayed and then disrupted, and then we ended up with only 29 patients. But fortunately, we are still able to do it anyway. And then the protocol, as well as the feasibility study, has already been published earlier. The device we use is one of the 3 or 4 most validated research-grade actigraphy called GeneActive. And the algorithm I use to analyze their sleep is also one of the most validated and well-used R package called GGIR, which has been validated against the UK Biobank, so Whitehall 2 study. And also, with significant amount of elderly patients, and also validated against PSG. So what I'm doing is I use this sleep data to correlate with the questionnaires-based data to see how different or how similar their sleep can look like. What I found is that, indeed, these patients sleep quite poorly, regardless of their gender or sex. As you can see, if we use a relatively lower threshold, so to see if they have any sleep disturbances, nearly all of them have some sort of sleep disturbances, as you can see here. And if we use a tighter threshold, using 85% sleep efficiency as the threshold, roughly at least 50% of them has some sort of sleep disturbances. But that is for those more general and more qualitative results. If we go to those more quantitative results, for example, if we try to look into when they started their sleep overall, or how long they sleep, when do they wake up, we can see overall, if we try to plot this Blend-Altman plot, which is a very common method used in sleep research to compare different sleep measurement. It's not too bad, and it's also not too bad if we average the results over two weeks period time. But you can see using this, if I do a regression analysis and calculate this formula, you will find that actually those X and Y are closest to each other once during the period between 9 p.m. to 10 p.m., which is roughly the kind of last service round provided by the care home caregivers. And also, if we look into their wake-up time, you can find that their wake-up time are basically not very, they basically, the device-based results and the questionnaire-based result are not very well correlated. Because this is not a large number study, so I have the opportunity to dig in to see what happened in every single patient. And I found, well, actually, using the device-based data, I can calculate this sleep regularity index, SRI, which has also been validated in large cohort. And SRI is another story, but basically it measures how regular you sleep, like what's the percentage you are in sleep during a period of time. And I found their sleep regularity index is very, very poor. I mean, you might not have an idea like what does 26 something means, but there is another study just published earlier this year in neurology. They found that in those patients with early stage dementia, their sleep regularity index is roughly 60. So 26 something is really, really bad. So really irregular. And if we further look into the difference between those two measurements, we can also find, although they have some agreement in this kind of, although they do not have a good agreement initially, but if we try to look into each patient and found that the reason why they do not have a good agreement, I can find there are specific patients who sleep quite atypically compared with other patients. For example, they sleep too short or sleep too early, too late. And if I try to adjust that, just to pick them up and compare them with what their results are in questionnaires and correct these results, the correlation will somehow be improved, but still not the best. Because I later found there are also some problems in actigraphy, because actigraphy cannot really tell what's the best time or what's the expected time for sleep. So if I write an algorithm myself to define an ideal time for sleep, this will further improve the sleep efficiency correlation. So what I'm trying to say is that, first of all these late-stage agitated dementia patients living in care homes, rather than what we think that, well, they are in care homes and they might be offered with a regular time-scheduled activities for them to live with, but actually they actually sleep quite bad and quite irregularly. And second of all, I found that, questionnaire-based results and actigraphy-based results, if actigraphy-based result average across a few days, in my study across 2 weeks' time, they relatively has a good enough qualitative, good enough agreement with questionnaires with regard to these qualitative variables, but not so good for those quantitative or longitudinal variables that can measure, for example, the sleep regularity index. And actually, sleep regularity index can only be measured with actigraphy or other wearable devices. And finally, I have to admit that this is a small sample size study using convenient sampling, so it must be interpreted with caution. But and also at the same time, because the STAND trial is not fully unblinded, so at the moment I don't know whether device-based measurement is more precise or those questionnaire-based is more precise. But I will argue, I will be very curious and I will be very excited once our study has been offered the blinding code, and I will be able to analyze, compare the differences. It must be something very interesting. And I think, hopefully, obviously, as you can see, although we don't know whether it's more precise or not, actigraphy or other wearable devices are capable of doing something more or more longitudinal and hopefully more objective. And that's all for my talk. And I'm going to talk about another study that has been collaborated with my Taiwanese colleague regarding wearable device on the therapeutic end in one of the Pint of Science activity a few weeks later. So if you are interested, you can scan the QR code and we can have a Pint of Science.
Thank you for having us here today. And before we begin, we would like to introduce ourselves. I'm Dishaa, and this is my colleague Nishat, and we are the central RAs for the research study running by University of Oxford, and we are supervised by Dr. Ivan Koichev, who's the chief investigator for the study, and Dr. Simon Yag, who is senior program manager at DPUK. And now then Nishat will introduce us to the study.
Thank you, Dishaa. So just before we begin to dive into the details of the study, I want to just begin with just briefly explaining what the FAST study is. So the study is a pilot study that investigates feasibility and acceptability of scalable biomarkers of brain health. And from this, we are working towards two main objectives. Firstly, to study the link between blood biomarkers and also dementia indicators. So blood biomarkers includes neurofilament light and also p-tau 217. And the second objective is to evaluate how practical and acceptable using blood biomarkers are. So the data for this is collected using feedback questionnaires and also to evaluate remote cognitive testing. And now looking into the, sorry, I forgot to change the slides. Now looking into the study methods and how we are achieving these aims. So currently the study aims to recruit 1,000 participants across multiple NHS sites. And currently we have just under 600 participants and with 16 actively recruiting sites. And participants are recruited using platforms such as Great Minds, which Dr. Vanessa touched on earlier, and also using platforms such as advertisements and referrals. We are also, in terms of the participants that we are recruiting, so we are recruiting cognitively healthy individuals, individuals with mild cognitive impairment, subjective cognitive impairment, and also dementia. In terms of the study process that participants are involved in, participants begin with completing an online consent form, which is either, so they can do it online, they can do it in person, depending on their preference. And they have a blood sample of 30 mils collected at 2 intervals, so at the beginning and also at 12 months into the study. They also complete a questionnaire just to understand the kind of feedback of the overall process of the study. This is also at the beginning and at 12 months into the study. And finally, they complete a remote cognitive test at 3 intervals, at the beginning, at 6 months, and also 12 months into the study. Now I'm going to hand it back to Dishaa, who will give a little bit about the analysis of the study.
Thank you. So here is the preliminary analysis, and this data is from the cognitive assessments. And just to understand the charts better, the green line here shows the average performance of the age match controls, and the shaded area in this diagram is how the FAST cohort performed on these tests. And as we can see, that the FAST participants performed worse than the age match control in 8 domains out of 12. And they were significantly impaired in psychomotor speed and cognitive flexibility, that is 2 standard deviations below than normal. And we can see it in red. They did perform normal in 2 metrics, which was easy condition of the object location memory task, also known as OMT, and relocating the target to its recalled position. There were no reliable differences across visits found so far, but this could be because a very small proportion of individuals have completed the visit 2 and visit 3 so far, as we are still recruiting. And here is the data from the blood analysis. As we can see, the p-tau 217 levels in the diagnosed versus undiagnosed groups. The levels in undiagnosed groups were lower than the diagnosed group, that is dementia and mild cognitive impairment. And this here is a graph to show the feasibility and acceptability of the study as a whole and the methods involved, that is blood collection and cognitive assessments. And as we can see here, 97% of the participants found the blood sample collection and the study as a whole acceptable, whereas 88% participants found the cognitive assessments to be acceptable, which implies that the majority of the participants are overall happy with the study and agree with the methods involved.
And now to conclude with the key messages on this study. So the study investigates correlations between cognitive changes and blood biomarkers in dementia patients. So it's starting with a pilot study and hoping to extend to a larger study in the future. And the use of blood biomarkers through the study is a really strong advantage, as it offers scalable and non-invasive approach to biomarker discovery. And these biomarkers provide several benefits, including early diagnostics, treatment monitoring, as well as targeted interventions in cognitive disorders. Before we end today, we would like to show our thanks to our supervisors, that's Dr. Ivan and Dr. Simon, for their endless support throughout the study, as well as Dr. Sidja and Dr. Masoud and the rest of the DPUK team. Thank you.
Thank you for that final talk. I think it's interesting that people found it more acceptable to have a blood test than a cognitive test. That maybe that says it all, or maybe that says something about the people giving the tests and what psychiatrists have been doing for years. I don't know, but there we go. Okay, we've come to the discussion section now, last session of the day. So if the panel, the speakers from the earlier talks would like to come and take a seat on the platform here, that'd be great. And I think we've got some questions on the Mentimeter to get you thinking about what you may want to ask us. So I think you should have QR codes on your tables, on the screen here as well, if you want to log into the platform for the first question. Okay, so these are the two questions which I hope you may have coming up on your screens now. So the first question is, what is the key change that you think we need to deliver in clinical trials now? Okay, straight away we've got quicker and speed. So speedy answers saying we need more speed. I think that's a good response. Diversity. Decentralization. It's an interesting response as well. So it looks like for a lot of people, the main point that's still coming across is about speed and diversity. Mathematics, I'm assuming that's maybe alluding to the potential for AI and algorithms. But widely applicable pragmatism. So the idea again that we need to be using real world data, I think, and real world environments to our advantage. And collaborative working, again, mirroring accessibility, I would say, and integration. So actually, I think compared to the questions we asked you yesterday, this is even clearer in terms of people's responses, because again, the more responses we're getting in, I would say, we're still reinforcing what we think we need is speed and diversity. Okay, we're thinking we've got all the answers in and we get a few more. Okay, we're going to move on to the next question, which is what do you think registries should deliver? So slightly different angle. What could registries provide at this point? So a bit more variation in response. I'm struck by the fact that access comes up in several of the responses there, and that immediately makes me think it's about access to the data, it's about access to populations for sponsors, commercial companies potentially, but also access for participants and patients and the public. And again, I guess it's kind of interesting that some people have flagged up the need for registries around phase 4 data. I think that came up earlier today, the fact that we haven't really had to think about that very much in terms of dementia drugs for some time, but certainly something that registries are going to be very vital in terms of supporting. Also, biomarker validation, again, mirroring everything we've been hearing over the last couple of days about the opportunities for novel biomarkers, but the fact that we need to be able to test them in as broad a population as possible. But again, main response here seems to be access and ease of use. So again, maybe mirroring some of the things that Robert was talking about earlier about the fact that while we would benefit from much larger data sets, including things beyond things like MMSE, actually, we just need to keep things simple so that we get as broad a data set as possible, a broader ability to be able to make these trials accessible to everybody. Okay, thank you very much for that. So we're going to move on now to questions for the panel. So any questions?
Good afternoon. Thank you very much for the wonderful talk today afternoon. Just have a quick question regarding preparedness. You know, what could be done or what has been done already to get the sites prepared? Because that is something which requires time. So if there are sites willing to participate in any of the trials, what could be done to prepare them in advance before you could set up the trial procedures? Thank you.
Victoria, do you want to?
Yeah, definitely. Is that working now? Yeah. Site preparation, definitely. So I think we are taking a lot of learnings from our primary care centres where they really have dedicated research hours where possible, and they are having research discussions with their patients as much as all the time. So research doesn't just come as a surprise to their patients. They know that their caretakers are going to find a study for them if possible. And I think that's something that if that could be replicated across more professionals, then we would not have this slow start to our enrolment phase. And there's definitely things industry can do differently. I know when we set up trials, we have a lot of systems and logins, and we're trying to innovate, we're trying to make things easier for the setup from our side as well.
Okay, thank you, Victoria. And I think it's an interesting point about having those discussions about research. I mean, I think personally that's one of the things that has really changed over the last few years. And I know I always feel it's because I'm seeing patients less in clinic than I was. So having the opportunity to have those discussions, you know, the opportunities are not there as they were. So we've got to think a bit laterally, I think, about how we get that information out on a regular basis. Other questions? There's a question here on the right.
Actually, I've got a couple of questions. One for Lynne. You talked about embedding patients in Biohermes 2 into commercial trials. Will they be allowed to go into non-commercial trials? And just as a controversial follow-up question, as a clinical trialist, I actually like to randomize patients in observational studies and turn it into a trial. Could you not take a generic repurposed drug? Now, I appreciate that might be controversial for companies, but you could implement a drug right now. I mean, I could do one for £2 a month with you, which would be perfectly worth testing. It just seems to me you're doing a lot of effort, and we can actually get an answer, a drug answer out of it as well.
Yeah, sure. Is this one on? Yes, certainly they can go into any trial that the investigator and the patient agrees is the best trial for them. And we want everybody to go into a trial if it makes sense, or to go on a database of screen failures. And certainly in our Ophelia trial, the 112 screen failures who didn't reach the amyloid threshold on the amyloid PET scan, they went into trials where they had to meet the Prizegiving AD test as an entry. So although we had 112 that failed Prizegiving, well, passed Prizegiving AD, but failed the PET scan, they all went into a trial where Prizegiving AD was the main entry criteria. I'm actually with Davos running a trial in Kenya using metformin as a repurposed drug. It's safe. We're getting used, the population used to running a clinical trial, training them in GCP. We're training ourselves in Swahili, which is interesting, and just letting them get used to it. And that's part of the Davos Alzheimer's Collaborative. And yes, if we can, as GAP, get a repurposed drug that somebody is willing to test and give to us, and we've had a couple, but they never went anywhere. But we'd certainly be interested. We wouldn't say no, put it that way.
Thank you very much. And if you don't mind me hogging a question just for Victoria, I wonder if you looked across your countries at the number of trials that each centre was doing. I don't know whether it's quite true for dementia, but certainly in stroke, UK stroke centres are doing 5, 10, 15. We're doing about 27 at the moment. If I've given up going to the US with our academic trials because they want to focus on 2 or 3. If you're only doing 2 or 3, it's much easier to recruit fast, although you will recruit less patients overall. If you're in the UK, you will recruit more patients overall, but less into an individual study. So is the problem in the UK that we're just very successful at recruiting, but not just into one subset of trials?
Yeah, and maybe it's because of the Lilly strategy that I can talk to, is that we do not want to overburden our centres with studies because our goal is speed. So we have our favourite sites, obviously, and we know that if we start to overburden them, they are no longer going to meet the Lilly expectations. So we are always looking for new sites. We are using the NIHR site identification tools. We're using our own, our team who are out in the field to identify new sites, because the Lilly strategy is speed.
Thank you. Other questions from the floor? There's one over here.
Hi. Hi. Sorry, a beautiful set of talks today. Great to hear all this progress in terms of, bigger trials and faster trials. And I think the general gist is, bigger and faster is better, but I have a slightly controversial proposition. So thinking about heterogeneity in patient groups, and we saw some evidence over the last 2 days that biomarkers increasingly show that there's diversity in patients and all these kind of things. So do we actually also need to go at the same time in the other direction? So what's your opinion, opinion on N of 1 trials, for example? Do we need to do close monitoring of one of a few patients over longer times with randomized treatments at individual patient level? Is that something that you're thinking about at all? Or is there anywhere in the plans for any of these platforms that you've presented?
I can talk about, I guess, just from a kind of dementia mission perspective, but kind of broader dementia research piece. So I think we can't take our eye off of that. And so I think, there's some really interesting research in that space. And so actually when we're talking about wanting to do sort of bigger, better, faster, that can't be at the disadvantage of other kind of smaller areas of research and trials that we need to then be focusing on as well. So I think certainly from some of the bigger programs from sort of the UK government mission, but also from Alzheimer's Research UK, we would be looking at some of those opportunities so that we don't miss out on some of the key questions that we'd need to address in this, because otherwise we don't have the sort of diversity of trials and participants either.
I would agree with that. And I think the other thing is we just need to be smarter as well about using the data that we have. I mean, we flagged out the fact that one of the bonuses for the UK, Victoria, I think you mentioned, was we have this wonderful data set sitting within the NHS for everybody, basically, but we don't often get to utilise that well in trials. There's a couple more hands up. James.
We've heard a lot about registries feeding into trials, but less about the reciprocal flow. What could be done to get people coming out of trials as themselves and their data flowing into registries? Would that not also be very helpful to speed up the cycle?
I've been involved in a couple of trials where once the trial finished, they did go into a registry. It's not unheard of. It depends at that point who's going to pay. And that was a sticking point for a few of them, why they didn't go into a registry, because do the pharma company get useful data out of it? They may not. If they don't think they will, they're not going to set it up, because you do have to monitor it. But certainly it's happened. It's not unknown. But there is a question of funding, and for how long will the funding last?
I'd say, I think that's something that we're certainly looking at. So I think as, the first generation of drugs start to come through, I'm looking at then some of the sort of next generation of trials. Actually, there is potentially some useful data, there's some useful follow-up. There's some, I think, useful follow-up in terms of our healthcare systems as well, around how those treatments are delivered, and a kind of conversation around where that needs to land and where that hosts. But I think there's a role for charities in that space.
Thank you. Ivan.
Yeah, great to see another great initiative with BioHermes in terms of a deep phenotyping a group of people that I'm sure in the short term will be fantastic for those trials that recruit them. I guess my question is, you know, from experience, these are, you know, if you like, trial-ready cohort of people who are post-diagnostic. These are cohorts with a relatively short lifetime, right? Because this is a progressive disease. We go through cycles where there's a lot of interest. I'm thinking of EPAD, the EPAD study. Now we're seeing some great initiatives. A lot of money gets put into essentially deeply phenotyping a sample of 1,000, 2,000 people. It's great scientifically, but in terms of feeding into trials, it's, you know, it doesn't seem to be sustainable because, you know, this is not a group that you'll be able to carry into trial for the next 10, 15 years. So I guess my question to the panel is how does the, if you like, the system of doing research and perhaps doing clinical work need to change in order to make sure that this is a sustainable pipeline so that we have people who are of sufficient level of characterization ready to go into the next trial in 5, 10 years' time, and how do we fund that?
Yes, I mean, I think that's part of what we're trying to do in the mission, which is essentially to work with industry to find the next generation of patients to come forward for trials and who are going to benefit from the new disease-modifying treatments. So completely agree. So I think, you know, we've seen where it hasn't worked historically and the reasons why it hasn't. So we need to learn from that. But there are certainly some opportunities, and obviously biomarkers give us an opportunity there. So looking at some of the initial work of the mission is going to be looking at those kind of disease-enabling biomarkers that allow us to think about who comes forward to which clinical trials when and how we find them, but also thinking about some of the other data sets that we have here. So things like our future health. Actually, are the data sets there that will tell us around, or what we can work with, that will tell us who those people and how we can then bring them forward to be in the right clinical trials at the right time.
Yeah, I think it's a great question there, Ivan, isn't it? It's kind of thinking about the next few years, but actually we need to be thinking about 10 years now. I'm really struck by the fact that a lot of brain health clinics, we haven't really referred to those much over the last couple of days, but these are service development initiatives, if you like, at a number of the sites that are involved in our networks and the DTRC, and looking at how we use some of the innovations around biomarkers, particularly to more accurately diagnose people earlier on. But I think a lot of the sites are thinking about how do we get the balance between data that's needed clinically to diagnose and offer a prognosis versus data that's actually going to be useful in the next 5 or 10 years to be able to funnel those people into trials. And I'm always struck by the fact that most people I see in my clinic, I think this is all happening already. They think they've usually signed some form at some point, maybe in primary care, that allows for their data to be shared. So they think we're doing this already, and this is very much what people want to happen with their NHS data. So now we've just got to make it happen practically, I think. Any final questions for the panel? Okay, well then I think just leaves us to thank you one more time, panel, and for your speech.
So I won't keep you long. My goal is to give you roughly half an hour of your life back, and maybe you might be eternally grateful for that. So, first of all, I think this has been a fantastic couple of days. I honestly want to thank you all for coming. I want to thank you for the quality of the questions and the grace of the questions as well. I want to thank all the presenters for all their fascinating perspectives and very interesting talks. It's invidious to pick out highlights, but I have to say that David Bennett was a force to be reckoned with, just going on and on. The next thing, the next thing, the next thing. Goodness me. So for him to come and join us, to come over here just for a 24-hour stop was a real privilege for us. That was great. Just a little bit of housekeeping, if I may. A little bit of courtesy, really. I would like to thank the DPUK team. I stand up here resting on a whole load of very capable people. Simon Young, who's effectively managed this whole thing. We've got a program which I shall refer to shortly, which runs very, very smoothly. I think we've had a lot of good comms stuff with Rod and team. I'd also like to thank the Congress Center staff. They have been polite and helpful, courteous to a fault. And I'd be very surprised if we don't come back, because the service has been so very, very good. And there were no fire alarms. Wasn't that wonderful? That's excellent. Good. So we will be giving prizes for the flash talks, but they will have to be decided later, because obviously we've only just had 2, and therefore it's not fair to judge them without having due consideration. I would like to just recap what you've been presented with. This is what we're talking about is DPUK 2. This is our second tranche of funding. And for most of our programs, we've achieved our goals within the first 3 years of a 5-year program. This is all credit to my associate directors, who have just made life so much easier for me. So there's James from Cambridge, and Susan from ARUK, Vanessa from Oxford, Simon from Swansea, and Ian from AstraZeneca. So they're dotted all over the place. And they are so easy to work with. They don't even correct my gaffes. You know, I know I've made them, but they're just so gracious, and I'm really grateful. Then there's the second tier of scientists that we're bringing on and maturing for the next generation. And I would particularly like to acknowledge the work of Sarah and Ivan in that. They've, fantastic initiatives they're taking. But what does all this work? What does all this deliver? It delivers a very mature data platform. We have I think, one of the best data platforms for research data in the world. It's extensive, it takes multimodal data, it has good service delivery times. It's very, very rarely down, and the staff are really helpful if you have a query. And that's extraordinary, and well done, the Swansea team. We have, I think, the largest UK dementia trials network, 52 sites integrating primary and secondary care, clinical and preclinical. It's remarkable. And the registers are almost 70,000 individuals, 20,000 of whom are genotyped, again, preclinical and clinical. And we have a dedicated experimental medicine program. Doing experimental studies or even mechanistic studies in people is hard, okay? And we're doing 3 themes on synaptic health, neuroinflammation, and vascular disease. And you'll work out why I'm going through all this in about 5 minutes. And we have a dedicated biomarker program through the readout. This is going to be revolutionary. It's so timely. I can only give credit to ARUK for pushing this through, for knocking on the door, getting partners in, and particular credit to Susan for doing this. We have global partnerships. Just for today, for example, and I have to write it down, we've had presentations from for the last 2 days, the Netherlands, USA, Germany, Norway, Sweden, India, Romania, Taiwan. And that's remarkable, absolutely remarkable. And we expect our network to grow into DPUK 3. Also, I think we're really privileged to have Hilary with us to present the Dementia mission. And what I want to say publicly is all of these assets I've been describing as DPUK 2 are completely at the service of the Dementia mission. We look forward to entirely supporting it so that this becomes a success and that we contribute to the transformation of dementia science in the UK. So much for DPUK 2. And I would like to encourage you to take these wonderfully glossy mags in there, which have got highly dubious pictures of myself in there, and please devour them. Take them as a reference. Because where we're going with this is something called DPUK 3. We have been invited by the MRC to submit a renewal. This doesn't mean to say that they'll rubber stamp it. It probably means that they will give it even deeper scrutiny by peer review than would otherwise happen. And that is absolutely the right thing to do. But for this renewal, we will be doing 3 things. We will be revising our structures so that our program can expand. We will be revising our partnerships to make them more flexible and more agile and of greater value to academic and industry partners. And we will be revising our programs and projects so that they are more aligned and more integrated and mutually inform one another. What we're doing today, the last 2 days really, is inviting you to contribute to that process. We will be writing a draft protocol over the summer period. We will then revise it. We will then submit it to MRC, probably later in the year, so that we can not only rebut all the criticism that we will receive from our very erudite peer reviewers, but we can then look forward to the program being renewed in January 2026. Okay, so we are genuinely inviting you. We would like to increase the globalization of what we do. We would like to give away our informatics to the global south. We would like to help train the next generation of dementia researchers wherever they may be. And that's probably not in the USA, primarily in the USA, or primarily in Northwestern Europe. Okay? But we would really like to do that. We would like to work out how better to work with industry. We would like to know how it can be so mutual that there's a very clear line between the point where there's greater public benefit in keeping the data within a company and then greater public benefit for sharing the data for all scientists to use. And there are criteria for understanding this. So we need to develop the data market so that it works optimally, efficiently, for the best good for society as a whole. And DPUK is committed to doing that. So I welcome you. I welcome you to give us your ideas, to come and be partners with us so that we can genuinely make this place a better place, this world a better place to live. Now, this conference wouldn't be possible, really, without our sponsors. And there they are. And again, I want to give due credit to them. They are really helpful to us. It's critical to the success of this program. But I also want to thank our funders, not our partners, our industry partners, who we value highly, and not our academic partners, again, who we value highly, but actually our core funders. And so I'm just going to read them out. So first of all, it's obviously MRC, and MRC have been really supportive of us. They recognize it's a challenging program, particularly with COVID, but they've been really helpful and supportive. I think I'd like to thank ARUK, we've got Susan, we've had Hilary here, we've got the readout, the Biomarker project. And again, just really, really helpful. And Alzheimer's Society also, we really greatly value them. And then we got to know ADDI, and that's been really helpful because the working relationship there is really encouraging. You know, our ADDI handler is Matt Clement, and he's a delight. And he's sometimes so polite. I don't know whether I'm being criticized or complimented. I just prefer that it's complimented, but sometimes maybe there are one or two things we need to adjust in our program, and he's very gracious in helping us do that. So I think those are all the thank yous. I'm extremely privileged to be able to lead this team and to have your generosity of time and thought as you input into this program. So thank you all very much, and come back next year.